Hi everyone, welcome to today's lecture. Let me just pull up my slides. Oh, let me share them first. All right, so today's lecture is going to be on the topic of phylogeography, which turns out to be many people's favorite topic of the entire semester, because phylogeography allows us to reconstruct the spatial and temporal movement of pathogens across landscapes. So we can learn about where different strains or different pathogens came from and how they arose. Um, but today's lecture is also going to bring together many of the statistical concepts that we've already covered in weeks 2 and 3, including how we infer phylogenetic trees, as well as how we model the evolution of character traits on those trees. So when we talked about Markov models of sequence evolution, we thought about how a given character trait, like a given nucleotide base, would evolve on a tree. And phylogeographic methods use many of the same ideas, but now instead of thinking about the character trait as, say, a given nucleotide or amino acid, we're going to think about that trait as being the location of a given lineage through time, where that location can actually be a given geographic location or maybe a given subpopulation in which that lineage resides. So as I already mentioned, phylogeography is really all about reconstructing the spatial and temporal movement of lineages across the phylogeny through time. And so it's very closely related to the idea of ancestral state reconstruction, which we brought up in week 1, where the idea is we would like to be able to infer or reconstruct where lineages or branches in our tree were at some point in the past, as well as where internal nodes in the tree, where those nodes might have been, whether that's a given location or a given subpopulation in a larger population. To motivate today's lecture, as well as the tutorial on Wednesday, we'll revisit the origins of one of the most famous plant infecting pathogens, Phytophthora infestans, which all of the plant pathologists in the class will know well, as it causes late blight on potato, as well as other solanaceous crops like tomato. And it's, of course, most famous because it caused this huge epidemic in Ireland and throughout the British Isles, and actually in the rest of Europe and North America as well, but mainly concentrated in Ireland, where the entire potato crop was wiped out for multiple years in Ireland and caused this immense famine. So people have postulated different sort of hypotheses about where this lineage of P. infestans first arose. One theory is that it arose in the Andes, somewhere in like modern day Peru or Colombia. And this is well supported biologically by the fact that the main host of P. infestans, tuberous solanaceous crops like wild potatoes, are native to the Andes, and so it would make sense that the pathogen arose here. Also, Gene Restano, who was in our department, in the Department of Entomology and Plant Pathology for many years, did this great genetic analysis in the mid 2000s where she sequenced both nuclear and mitochondrial genes of this pathogen, and reconstructed phylogenies or coalescent trees of these different genes, and then used those trees to reconstruct where the root location was of these different trees. And most of these trees supported an origin of this pathogen in the Andean region. Um, and the analysis was interesting because it also implied that how the pathogen may have ultimately gotten to Ireland and the rest of the British Isles was sort of through this route where it came up from South America into North America and then spread across the Atlantic to Ireland. Um, but time went on and other people started collecting more and more genomes of P. infestans. including this group led by Erica Goss, who's now at the University of Florida, and Nick Grunwald at Oregon State. And they sampled many more genomes of P. infestans throughout Central America and Mexico. And then from reconstructing phylogenies of this new genomic data set, they ultimately came to the conclusion that the origin of P. infestans or P. infestans probably arose somewhere in central Mexico. And again, this makes good biological sense because there's diverse sexual populations of P. infestans in central Mexico, suggesting it could be like the source population for a bunch of genetic diversity of the pathogen. And many tuber-bearing Solanum species also live in this central part of Mexico. So I don't really have a horse in this race. I don't really know if the pathogen arose in the Andes or central Mexico. The interesting thing here is in the tutorial on Wednesday, we'll get to apply our phylogeographic methods and see how much confidence or certainty we should have in either of these proposed origin stories. All right, so that all begs the question of how do we actually do this type of phylogeographic inference? How do we reconstruct the spread of pathogens through time based on only our knowledge of where pathogens were sampled at present? Well, it turns out that there's two main flavors of phylogeographic analysis. There's what we'll call discrete trait models, which think about the movement of lineages between different populations as very much discrete, like instantaneous jumps between different locations or populations. And then there's these continuous models, which think about the movement of lineages more as a diffusive process where lineages sort of just gradually, slowly move continuously across landscape. And towards the end of today's lecture, we'll talk about which of these models is more appropriate for different types of pathogens. But for right now, we'll just start off with these discrete trait models because they're more broadly used, and this is what our tutorial will be based on Wednesday. So these discrete trait phylogeographic methods, or what I'll just call DTA models, are very similar to the models that we already saw used when we model sequence evolution. They are another example of a continuous time Markov process or Markov chain, where the rate at which a given lineage moves between any two populations is determined by this migration rate matrix M, where each entry in this migration rate matrix M gives the pairwise rate at which a lineage would jump between two different locations or populations. So, for example, M12 here would represent the rate at which a pathogen moves between location 1 and location 2. Almost exactly the same way we think about these Markov models of sequence evolution, where these rates would determine the rates at which mutations or substitutions happen between different nucleotides or amino acids in a sequence. And because of the similarity to Markovian models of sequence evolution, sometimes people refer to these phylogeographic models as mugation models, as sort of a combination of migration and mutation. These models have some nice mathematical properties, as we already saw when we thought about Markov models of sequence evolution. The thing that makes them really nice is that given the location of lineages at the tips of the tree, which we can actually observe, computing the probability of a lineage being in any state at any given time in the past is relatively easy. So if we think about the location of one of these lineages in populations, either 1 or 2, we can describe that as a vector, where here the probability of this lineage being in state 1 at time 0 will be 1 because we know it was in that population. But we can think about this vector of probabilities going backwards in the past and ask, well, what is the relative probability that it was in population one or two at any point in the past? To do this, all that we would need to do is be able to exponentiate the migration rate matrix and multiply it by the time that has elapsed since we observed the tip lineage. And that will give us a transition probability matrix that we can then multiply by the initial vector of probabilities. And so we can update our initial probability vector by this matrix of transition probabilities to get an updated vector of probabilities that represents where a lineage is at any given time, probabilistically where it is at any given time in the past. And so this is mathematically easy to do and computationally fast, which is one of the reasons why these discrete trait models have caught on. Um, but we've been talking a lot about Bayesian phylogenetic inference, and so how would you do this in a Bayesian context where we're using something like MCMC to infer our trees? Well, in this case, if we're using MCMC, what we can do is at each iteration of our MCMC algorithm, we'll sample a given tree, and then for all of the nodes in that tree, we can calculate the probability of that node being in each population using the matrix algebra approach that I just described. And then we can randomly sample a location for each node based on their probabilities. And this is nice because then if we continue to sample trees and their associated reconstructed states in our MCMC chain, what we'll wind up with is a set of trees with ancestral locations that we can use to infer the posterior probability of a lineage being in any given location. So what we'll wind up with is for any given node in the tree, we can look at how many trees we sampled where that node was in that particular state. And that proportion of time that that node was reconstructed to be in a given state will be proportional to the posterior probability of that node being in that state. So it's easy in this case to use MCMC to infer the relative probabilities of lineages being in a given state, which we often need to do because there's often an incredible amount of uncertainty that comes along with inferring these ancestral states, especially if we're trying to infer where lineages were long ago in the past, depending only on data that we have about where these lineages were at present. So being able to take into account full uncertainty in not only these ancestral traits, but also our phylogeny is very helpful. Another nice thing about these DTA models is that they scale well with the size of data sets, so they can be applied in situations where you might have hundreds or even thousands of samples or tips in a tree. And they even scale pretty well, even if we have a large number of possible states or ancestral locations at which a given lineage could be. So this is a nice example from Philippe Lemay's group. And Philippe was like one of the main leaders in this field of phylogeography and developed many of the methods that we'll talk about today and the tutorial on Wednesday. But in this case, Philippe and all were interested in reconstructing where avian influenza or avian H5N1 arose based on samples that they had collected from all over the world. And so in this case, they were able to consider, hundreds of samples collected from all over the world, as well as many possible sampling locations, and then infer which one of these sampling locations represents like the most likely origin or ancestral population of these viruses. So in this case, they were able to reconstruct that either Hong Kong or Guangdong most likely was the root location of these sampled H5N1 lineages, suggesting that The origin of modern avian influenzas might be somewhere in East Asia. Of course, as we talked about in the very first lecture of the entire semester, um, assuming that the root of a given tree necessarily is representative of where a pathogen first arose, can be problematic and can mislead our inferences. So we always have to interpret these inferences about the origins of different pathogens with appropriately. Um, and so one thing that I would like to point out is that there's many reasons, or at least four reasons that I can think of why inferences about the root state in a given phylogeny might mislead us when we're thinking about the origins of a given pathogen. Um, the first case is sort of the easiest to understand, and that's we simply might have not have sampled individuals from the true source population. So in this case, a pathogen truly arose in the red population, but we only sampled lineages from the blue and yellow populations. So we have no ability essentially to reconstruct the true red population as being the true origin. Another thing that can happen is we might actually sample the true origin location, but depending on exactly which lineages we sample, we might just get unlikely and we might infer the wrong or reconstruct the wrong root state, even though we have samples from that population. This would happen especially if we've undersampled from the true location, because then samples from the true root location might be sort of nested within clades of samples from other locations, which could mislead us about the likely location of the root. Um, Finally, something that I think people often neglect to consider is that if we are only sampling modern day samples, those samples might be completely non-representative of the historical geographic distribution of samples, and so might mislead our inferences. So if, say, at present day, a pathogen is, mostly concentrated in the blue and yellow population here, we would probably reconstruct that the root location is in the blue population. But that's might just be because older lineages that were in other populations have had time to die off or simply just weren't sampled. And this is actually likely what happened in the case of Phytophthora and Fastans, because if you take modern day genetic diversity of this pathogen and reconstruct where P. infestans came from, you only get as far back as sort of like the 1920s and 1930s in the US. Um, and so that actually, of course, does not explain at all where these lineages that originally caused the potato famine came from. We would have had to sample older lineages that are now extinct in order to reliably infer where the root location is. And one further complication is it says that there could have been multiple introductions into a given host population, um, such that the actual origin of a pathogen might have been much farther in the past, and the different introduction times are only representative of the times at which a subset of the pathogen population moved into a given host population. So inevitably, people will still want to reconstruct the ultimate historical origins of their favorite pathogens. So they still use these phylogeographic methods to reconstruct origins. Um, and I don't feel like they often give appropriate caution when interpreting these inferences. Or maybe a better way of putting that is they overinterpret the inferred root location as being truly representative of uh, the origin or source of a pathogen, when perhaps we should be placing more caution or more caution in our interpretation of where these pathogens came from. All right. Another problem that arises with these phylogeographic models is that they have a lot of parameters, especially as the number of different populations or spatial locations grows. So if we just let n represent the number of possible locations or states in our model, we would have to infer this migration rate matrix representing all of our migration rates between all of these different populations. And if n is small, say n is 4, that's only like a 4 by 4 matrix. And we don't have to estimate the diagonal elements on the matrix because these are assumed to be 0. So we have about n times n minus 1 uh, parameters that we infer, and for small n, that's very reasonable. But what if we had like 40 different populations? Then this leads to truly massive migration rate matrix. So for n equals 40, there'll actually be close to 1,600 migration rates that we'll have to estimate. Um, and that's problematic for a few reasons. One is sort of just like the classic statistical overfitting problem of if we have all these additional parameters in our model, we don't think that we'll be able to reliably estimate them all. And two is just that if we think about it, with finite data, a limited number of samples, it's actually unlikely that our sample will actually contain migration events between all possible pairs of population events, meaning that nowhere in the history of our sample was there actually a migration event between any two populations. So it's worse than just overfitting because then we'll be essentially trying to estimate migration rates from data that contain no information whatsoever about these migration rates. So we have to have some way of dealing with this what we call cursive dimensionality or having too many parameters in phylogeography. And the clever thing that people have come up with, and we'll see this when we do our phylogeography tutorial on Wednesday, is that a favored method has become to use this idea of Bayesian stochastic search variable selection to try and identify which elements of this migration rate matrix are actually important to explain the data. So Bayesian stochastic search variable selection is a bit of a mouthful, so I'll just say BSS VS. But the main idea behind the method is quite simple, and that's we want to take this matrix of all possible migration rates and sparsify it, meaning that we want to determine what small subset of these rate parameters is actually important to explaining our data. And the way that we do that is in front of each rate parameter, the m's in the model, we put a dummy or indicator variable, which I'll call delta ij, where the I and j correspond to the corresponding migration rate. So delta 1, 2 corresponds to the migration rate for a population between populations 1 and 2. And these indicator variables or dummy variables can take on just one of two states. They can be either one, in which we can think about that migration rate kind of being on in our model, or that dummy variable can be 0, in which case that migration rate will be 0 and there'll be no possible migration between that particular pair of populations. So then the big idea behind this method is that we know that most of these entries in this migration matrix should be 0 or pretty close to 0, so not important at all. And we want to just figure out which subset of variables or migration rates is actually important that has migration rates significantly higher than 0. This is also nice because it allows us to do a form of Bayesian model selection. So model selection inevitably arises when you have competing or different models and you want to figure out which model better explains your data. And there's many different model selection criteria. in statistical inference, people have used things like uh, AIC or KIKI information criteria if when doing likelihood inference. There's also the Bayesian equivalent of AIC called BIC, or Bayesian information criteria. But what's popular in phylogeographic, um, in the world of phylogeography, is to use model selection based on Bayes factors. And as we'll see, Bayes factors have this nice interpretation that they allow us to think about how much probability or how much support we have behind two alternative models. In this case, when we think about alternative models, basically what we're thinking about for each possible rate parameter is two possible models. Uh, Model M1 that contains a non-zero rate for a specific migration rate, say between populations K and L, and model 0 that assumes that that same rate is therefore 0. And so we can think of model 0 as being sort of a special case or reduced version of model 1. And then the nice thing about using these BSS-VS methods to do model selection is that we can actually then use MCMC to actually calculate our Bayes factors for us and see how much support or probability there is for a model that either does or does not contain a given migration rate parameter, which is the main way people have come to sort of like quantify how much support there is for the inferred migration rates that people estimate in these from these phylogeographic models. So what a Bayes factor actually is mathematically is it's basically a ratio of how well the data is explained by one model over how well the data is explained by another model. So this ratio here is actually a ratio of two likelihoods, or to be precise, a ratio of two marginal likelihoods, because they consider the probability of the data arising under either model, considering all possible parameter values of that model, which is what makes it a marginal likelihood. But the interpretation of these Bayes factors is really straightforward. Basically, we can think about it as a posterior odds for preferring one model over another. So if a Bayes factor is 2, we would know that the probability of the model 1 being correct over the probability of model 0 being correct is approximately um, one half, or sorry, is approximately twice as the probability of model 1 being correct is twice as high as the probability of model 0. So people have very standard sort of like guidelines for how you would interpret these Bayes factors. Generally, Bayes factors between like 1 and 3 are considered to be weak support, because those are relatively small odds that one model outperforms the other, whereas Bayes factors above 3 and certainly above, say, like 10 or 20 are considered to be strong evidence that one model fits the data better than another model. And the beauty of doing this all within this Bayesian stochastic search variable section is that in our MCMC framework, basically these dummy variables that we're using, these indicator variables, the deltas in our models, they'll be sampling different values between 0 and 1, where 1 represents sort of the on state and 0 represents the off state. And so if we want to compute the Bayes factor for a given model with a given migration rate over a model that assumes that that migration rate is 0, we can look at the proportion of the time the model spends in sort of the on state where the dummy variable is 1 relative to the portion of the time in which it's in the off state, which will just be 1 minus that p delta. And then by calculating this ratio, we can then see how much posterior support we have for the model that includes this rate parameter over a model that does not include this rate parameter. And if all goes well, then hopefully what we'll wind up with is instead of having this huge migration rate matrix um, with possibly hundreds or even thousands of migration rate matrices are indicator variables, and the Bayes factors that we can compute from those indicator variables will essentially tell us which subset of migration rates is actually important um, and actually has a high probability of being greater than zero. And so we would actually then need to include those migration rates in a model to explain the location of the tip locations that we've sampled individuals from well. Um, but yeah, we'll get more experience looking at Bayes factors and how to interpret them in the phylogeographic tutorial on Wednesday. Um, so oftentimes people think about phylogeography as, simply a way of doing ancestral state reconstruction and reconstructing the origins of particular lineages or inferring where a particular one lineage came from at some point in the past. But a nice development that's sort of arisen in the field of phylogeography over the past decade or so has been that we can actually even go a step farther. And instead of just inferring where lineages were in the past, we can begin to understand what sort of drives these migration parameters in our model, or what drives spatial spread between different locations. And one of the main ways that people have done this is by using these generalized linear models, which are just a special case of basically linear regression models. So in this case, we start off with something that is essentially exactly analogous to a linear regression problem. We say for each migration rate matrix, or sorry, each migration rate in our parameter, in our model, um, we want to know which explanatory variables can sort of like explain or predict that given migration rate. So then what we have then are a series of explanatory variables here, the x's, and we want to infer the relative sort of importance or predictive power of these different explanatory variables. So we infer the regression coefficients, betas, which tell us how important each one of these covariates is to a given migration rate. And just to give you an example to make this more concrete, these covariates could be a number of different things, but they could be something like, the actual physical distance between two locations. They could be environmental variables like the temperature or humidity in the different locations. Oftentimes when people are fitting these models for human pathogens, the explanatory variables have to do with the population size at both sort of the source and destination variables, because we think that, on average, there is more movement of humans between uh, locations with larger population sizes, and so migration rates might be well predicted by the relative size of the source and that source and destination location. And what makes this a generalized linear model instead of just a linear model is we can apply some nonlinear transformation, the G, wrapping up the additive linear part of this function here. So why this is useful is, say you have some trait or some explanatory variable like the distance between two locations that you think might explain um, the migration rate of a pathogen moving between those two different locations well. But maybe it's not a linear relationship. Maybe the distance um, sort of the migration rate decreases between two locations based on sort of like the log of the distance between them. In that case, you can have one of these GLMs where you have some appropriate transform function that accounts for that nonlinear dependence of the migration rates on our predictor variables. Um, a really nice example of this was uh, applied to the movement of seasonal flu viruses around the globe. Um, so in this case, it was Philippe Lemay's group again. They wanted to understand whether uh, passenger air traffic explains how quickly different strains of influenza can move around the world. So what they were able to do is they first did a phylogenetic reconstruction of how strains sampled from different parts of the world are related to one another. And they used one of these GLM models to then estimate the importance of different explanatory variables to the migration rates at which these lineages move between different locations. And what they found, um, not so surprisingly in hindsight, is that the best predictor of the migration rate of a given lineage of fluid between two different locations is basically how much passenger air traffic flow there is between those two locations. So this makes sense, if you think about there being two airports that are like hubs, say like Atlanta and Frankfurt. If there's a high volume of air traffic between those two locations, we also expect that because there's so many people moving between those locations, flu viruses will also be rapidly moving between those locations as well. And so the migration rate for the virus will be very high between destinations where there's more air traffic. What's also sort of interesting about this analysis is that it shows that unlike passenger airflow, the actual distance between airports is a very poor predictor of migration rates for the virus. Again, here they're using these Bayesian stochastic search variable selection to calculate the probability of how often these different variables are included in the model. So here you can see that like the average distance is only very rarely incorporated into the model, and its effect or regression coefficient on the actual migration rate is very small. It has basically effect close to zero, whereas passenger flow has a very large effect on the migration rates. So this actually turns out to be a common theme, at least for human respiratory viruses, where the actual physical distance between locations is often a very poor predictor of how quickly um, pathogens will move between different locations. So here you can see basically a plot showing how quickly two different pathogens, influenza H1N1, which was the pandemic strain in 2009, as well as the original SARS coronavirus from 2003, um, how quickly these viruses spread between different locations around the world based on the distance between those different locations. And here you can see that actually there's only a very weak positive relationship between the distance between these locations and how long it takes these viruses to move between these two locations. Whereas if you take the passenger airflow between uh, the air traffic passenger flow between two different locations and calculate what we can think of as an effective distance that accounts for how much air traffic there is between two locations, such that the more air traffic is, the more closer these locations will be in this sort of effective space. We can see that these effective distances based on air traffic very precisely predict how long it will take a virus to go from any one location in the world to any other location in the world, which is kind of cool. It suggests that, well, first off, that we're getting genuine insights into the spatial epidemiology of these pathogens from genomic data, that we can actually learn what factors sort of predict the spread of these pathogens from genomic data. but also two, that there's like underlying hidden, what we can think of as like hidden regularities to how these pathogens are moving around the world. And if we can learn what these predictive factors are, then we'll be able to much better predict how pathogens will spread spatially around the world. Of course, uh, these human respiratory viruses are a special case because uh, in this case, we sort of like understand how humans move around the world. That in modern times, that is mainly by air traffic, and so we also understand then how human viruses move around the world. But this is a lot less clear for many other types of pathogens, right? Especially when we start thinking about plant and other agricultural pathogens, where it's not so clear what actually predicts the spatial movement of these pathogens across complex, sort of divided landscapes. So how do we learn what's driving the spread of these agricultural pathogens? What can we use to predict their spread? Um, One really nice example of people applying these phylogeographic methods to plant pathogens to see what's sort of like driving their spatial spread was this nice paper by Travau and al. From about 10 years ago, where they wanted to understand how rice yellow mottle virus was moving throughout Africa. And so rice yellow mottle virus is a major single-stranded RNA virus that's become a real problem in rice production in Africa, which is important because rice is a major staple crop, um, feeding a lot of people in Africa. So these guys wanted to understand, what factors were driving the spatial spread of RYMV. And so they applied many of the approaches that we just talked about. They used these discrete trait phylogeographic models to reconstruct a phylogeny with ancestral states of where all these lineages were in Africa through time based on where they were sampled at present. And then they were able to reconstruct these movement networks between different locations where here, again, they're quantifying how much movement there is between different locations based on base factors. So arrows connecting locations with high base factors means the data is strongly supporting that there is a non-zero movement rate between these two locations. And so based on these base factors, then we can see that there's essentially two different subnetworks here, right? We can see that there's a high amount of connectivity in Western Africa, especially along the sort of Atlantic coast. And that there's this well-connected network of movement in Eastern Africa as well, but there's not much movement throughout Central Africa. And if you think about this for a second, this actually makes complete geographic sense because this part of Central Africa is very heavily forested and mountainous, and so not even animals like elephants and leopards move very easily across this landscape. And essentially, there's no rice production in this region of Africa either. So to more definitively sort of quantify this result, Travau and all also thought about how they could take distances, but instead of thinking about distances between different locations as sort of standard geographic distances, they were also thinking about distances in this sort of with this idea of using effective distances, where locations that are better connected through trade routes or through agricultural connectivity would have shorter effective distances between them. So what they did is they looked at sort of complex landscapes like this, where there's rice production, but it's broken up by many other pieces of land where there's no rice production, and they actually tried to quantify how connected the rice production systems were. And to make sense of this, you can kind of think of it as an analogous, these effective distances and analogous is to like a electrical circuit network, right? Where in an electrical circuit network, there's many paths connecting any two locations, like the blue with the green or the blue with the red. Um, but some of these paths through the circuit network might have high conductivity or low resistance, while other paths might have low conductivity or high resistance. And so, by way of analogy here, what they're thinking about is the sort of high conductivity parts of the landscape are parts of the landscape that have a great deal of rice production. And so there would be therefore like high connectivity through the landscape in these parts. Um, but this is complicated because there's many different ways that pathogens can move throughout these different pathways on the landscape. So what they do is basically they sum over all possible pathways weighted by their relative resistance or conductivity, and map that onto a simpler distance that quantifies the effective distance between two locations in terms of how well connected we think these populations should be. Um, and by sort of translating the agricultural landscape into these effective distances, what they are able to do is quantify how different predictors of spatial spread on these landscapes, how much that actually leads to higher resistance or lower connectivity in the landscape. And here they found that there's two main factors that sort of predict how well connected different locations are. Um, First of all, low connectivity, meaning fragmented landscapes where rice production is actually like physically broken up across the landscape by other non-rice producing regions, is a very strong negative predictor of ROYMV movement, which makes sense. And also production intensity, how much rice is grown at each one of these locations is also a strong positive predictor of movement throughout Africa. So taken together, I think this makes a lot of sense, right? Because essentially what we're learning by thinking about distances on this landscape, not in terms of actual strict physical distances, but effective distances that are quantified that measure resistance or connectivity throughout the landscape, we can actually see that how well connected the rice growing regions are and how much rice is actually grown at each of these two locations is actually a very good predictor of how much movement there is of our YMV between different locations, which makes sense. Before we move on to continuous phylogeographic methods, I would just like to note that, strictly speaking, phylogeographic methods can be applied to other sorts of traits beyond just geographic locations. In other words, the states or the subpopulations in these models can represent different things. For example, a common application of phylogeographic models when applied to pathogen populations is oftentimes the different states will represent hosts within different sort of risk groups or subpopulations. And another popular application is to think about the different states or populations as being different host species. So in that case, then people are able to reconstruct the ability of pathogens to move between different host species. And a really cool example of that was by this paper by Daniel Streicher that came out in Science a few years ago, where for bat rabies viruses, they actually wanted to see how frequently these rabies viruses were moving between different bat species in North America. And so by applying these discrete phylogeographic methods to these bat rabies viruses, they were actually able to reconstruct how much movement there, how often these viruses were moving between different bat species. All right. We're going to spend a lot less time talking about continuous phylogeographic methods just because they're less broadly used. They're more computationally intensive and just sort of like harder to fit to real data, but they can be very useful in certain contexts. So why would we use a continuous phylogeographic model over a discrete one? People often use these continuous models when pathogens diffuse more slowly across a given landscape. So we already said that these discrete trait phylogeographic methods that we just discussed assume that pathogen lineages basically instantaneously jump between different locations. Well, how instantaneous that jump occurs, of course, is somewhat arbitrary. I mean, in some cases, someone getting on a plane and going from New York to Paris is basically more or less instantaneous movement of a virus between those two different locations. But if movement between different locations is actually much slower and occurs on a much longer scale because the hosts actually move across a landscape in a much slower manner, then it might make sense to think about the spatial movement of these pathogens is more of a diffusive process where the host physically has to like move through the landscape. And these are most often applied to wildlife diseases. Um, it's like sort of two classic examples of these continuous phylogeographic methods being applied to West Nile virus in the United States, which is spread, it's a vector-borne virus, but also spreads through the bird population. And so birds can obviously move pretty rapidly, but birds can't get on, a cross-continental flight and fly from New York to LA. They much more slowly migrate across the landscape. So in this case, the movement of the virus across the landscape is much better captured by sort of a continuous diffusion process across the landscape. And the same thing for raccoon rabies in the Northeast United States. Here, the movement of the virus is actually limited by how quickly raccoons actually move across the landscape. And so raccoons don't actually move that quickly. And so it's taken multiple decades for rabies virus spread by raccoons to actually spread from its origin somewhere in Maryland up and down the Atlantic coast. So there's no hard cutoff that determines when to use discrete trait models and when to use continuous models. You kind of have to use your best judgment and your knowledge about the biology of these different pathogens to reason whether it's more reasonable to assume that pathogen lineages are jumping instantaneously between different locations, or if they're moving slowly and more diffusively in a wave-like manner across a landscape. But the basic idea of these continuous spatial phylogeographic methods is similar in some sense to the discrete methods. We assume that we know the locations of tips or where samples were collected. And then from those tips, we want to reconstruct the ancestral locations at the internal nodes of the tree, where now these ancestral locations could be represented in terms of where these lineages are in 2 dimensions. Um, and then if we know basically the ending location of where a lineage wound up and where it started, then each branch or lineage in our phylogenetic tree will basically represent a vector that tells us where a given lineage started and where it wound up. And if we have a time calibrated tree, we'll also know how much time elapsed from the beginning and end of that branch. So that allows us to then compute basically how long it took for a given pathogen lineage to move between two locations on a landscape based on how much time has elapsed along the branch. And so we can use these phylogeographic methods to compute useful things. One thing that you can estimate is what people refer to as the wave front velocity, or what sort of the maximal rate at which a pathogen is spreading across the landscape. So this would be like thinking about it as sort of a diffusive wave-like process. This would be how fast is the very sort of the farthest extent of the wave spreading across the landscape. And so by wavefront velocity, what we're measuring is how quickly are the fastest moving pathogens moving across the landscape. But we might also be more interested in sort of an average measure of how quickly, your average pathogen lineage is moving across the landscape. And so people oftentimes estimate these mean diffusion coefficients as well, where these mean diffusion coefficients tell us how rapidly sort of your average lineage is moving in space behind the farthest possible extent of the spatial wave. And so people have applied these methods to many different pathogens, but the West Nile application is perhaps the most famous because it's just a really nice example of how these methods work. So, based on the reconstructed relationships between different pathogen lineages, we can kind of see how the West Nile epidemic spread across North America. It started somewhere in New York in the very early 2000s, and then sort of like rapidly spread down the East Coast or the Atlantic coast, and then slowly started spreading across the continent westwards, sort of more or less taking a southern route. And so here, Pybus and all were able to estimate what the wavefront velocity of this pathogen was, in which case, by sort of plotting the farthest extent that the epidemic reached in its westward expansion along time, they were able to estimate how quickly the wavefront was moving, and sort of at its peak, shouldn't say peak, at its fastest rate, more or less, West Nile virus was moving approximately 1,000 kilometers per year, which makes some sense because while the virus is vectored by mosquitoes, it can be transmitted by birds as well. Obviously, birds fly around, and so it seems totally reasonable that this virus can move 1,000 kilometers over the course of a full year. They could also estimate these diffusion coefficients, which again captures sort of like how much on average, how quickly on average the area behind the wavefront is sort of filling in. And they did this assuming both that all lineages were moving at a homogeneous rate across the landscape, or allowing for heterogeneity or variability in this diffusion rate across different lineages. And what you see here is that if you account for this variability in diffusion rates, you actually estimate that there's a wide range of different diffusion coefficients. And some lineages, like the lineages that spread very rapidly down the eastern seaboard, were able to move very rapidly and then sort of got stuck as they moved farther and farther west. But then once the virus got to the Intermountain West, it was able to sort of move rapidly again throughout the Intermountain West. And the thought here was that this seems biologically plausible because both the Intermountain West and the Atlantic coast represents sort of avian flyways where birds rapidly fly up and down in these regions, and so are able to rapidly move the virus from north to south or north or south to north. But the westward expansion of this virus was more impeded. A subsequent analysis that was done by Simon Delacour and all sort of took a closer look at this data set and asked, similar to what we saw with the rice yellow mottle virus, well, what features of the landscape or what explanatory variables in the landscape can actually sort of explain or predict how quickly West Nile virus was able to diffuse across the landscape. So here they looked at multiple different explanatory variables, including things like elevation and forest coverage and how much grasslands there were, as well as more like physical things like annual mean temperature and annual precipitation. And what they were able to do is they were basically able to convert all these spatial variables into a raster file, and then use this raster file to basically predict what would be the effective distance between two different locations, much the same way as Travalo and all did in the rice yellow model first instance, um, where they wanted to infer whether each one of these variables had sort of a um, facilitative property and allowed the pathogen to diffuse at a higher rate. Like did any of these variables increase the conductivity at which these viruses can move through the landscape, or which one of these variables were associated with higher resistance across the landscape. And what they found was kind of interesting. They found that overall, the only explanatory variable to significantly predicts how rapidly West Nile virus is able to move across the landscape was mean annual temperature, which is a bit of a head scratcher at first, but one of the reasons why this may be important is if we think about the mosquito vectors, the warmer the temperature is, the more rapid the virus will be able to grow in mosquitoes, and therefore the what's known as the extrinsic incubation period, or how long it takes for the mosquitoes to incubate the virus, is shortened in warmer places. And so that might explain why warmer temperatures facilitate the movement of the virus, and also why it seems like that the way the virus was able to move across the continent was mainly through this southern route where there's warmer temperatures. All right. Um, so that wraps up our lecture on phylogeography. Um, I think what we've seen is that we've gone from a world where people were primarily interested in using phylogeography to reconstruct the origins and ancestral locations of different pathogens, including important pathogens like Phytophthora, um, which people are still very much doing. But now phylogeography in recent years has also given us the ability to sort of quantify what factors are driving the spatial diffusion or spatial expansion of different pathogens, both human pathogens, as we saw with the air traffic example of seasonal influenza, as well as agricultural pathogens like rice yellow model virus, where people have been able to actually link up sort of what spatial or environmental variables actually explain how quickly viruses are able to diffuse across these complex landscapes, which is exciting because then we can actually learn about like what factors are actually important for driving the spatial spread of these pathogens. And so on Wednesday, we'll take a closer look at the discrete trait methods, and apply them to our motivating example of Phytophthora, and you'll get a chance to actually see for yourself how much evidence there actually is for a Andean versus a Central Mexican origin of Phytophthora, which is fun. And if you're interested in the continuous phylogeographic methods, because you're working with a pathogen that moves in a more wave-like or diffusive manner across a landscape, there'll be a link in the tutorial to other tutorials that people have made for the continuous phylogeographic methods. All right, well, that wraps up the lecture today. If you have any questions, feel free to ask me during the tutorial on Wednesday, and I'll see you in a couple days.